Welcome student. In the next lecture of this week, we have started discussing about different plant parts and we have discussed very important plant organ, that is the stem. In this we will discuss another important plant organ, that is the root. Now root is basically an underground part, okay, like stem was basically found on above, root is found generally on the below ground, okay, and the main function of root is to absorb mineral and water from the soil and transport it to other parts, like transport it to stem, and then it will take to the other parts of the plant. And another important function is anchorage of the plant in the soil so that it can stand firm. Now, two types of root you all would have heard of or you would have studied, tap root and fibrous root, and on this basis we can also divide monocot and dicot, like there are different morphological features also on which you would have studied that how to differentiate between monocot and dicot. So this is also one feature, like dicot plant has tap root system, whereas monocot plant has fibrous root system. It is also known as adventitious root. See, there are many other differences also that I think so you all might have studied how to differentiate a dicot and a monocot plant. See, dicot have two cotyledons, that was the first important thing that you all would have studied, and monocot have one cotyledon, like in pea, in pulses you would have seen two cotyledons, and in rice and wheat just one cotyledon. Similarly, root also there is a difference in venation pattern of leaf. Also you would have seen that dicot has net like venation in the leaves and fibrous root has parallel venation, okay, parallel venation. So coming back to here, this tap root system, this originates from radicle, means any root which is originating from radicle is the tap root. See this, like during embryo growth, see it's just a rough diagram to just make you understand. See, you know that when cotyledons, you have seen an embryo like structure, you would have tried to split a pea seed or a gram seed, and you would have seen that between the two cotyledon a small structure is there which is actually embryo, and the above one is known as plumule and the lower one is known as radicle, and this radicle will give rise to root and plumule will give rise to shoot. So this root which is coming out from radicle is known as tap root, okay, whereas we call it a fibrous root when it is coming out from any other part of the plant, like generally they come out from the base of the stem or even from leaf. Also you would have many a times observed that some leaves, it's not very common feature that some leaves might produce some adventitious root like structure. So this is also fibrous root. Now tap root also, it has one main root, okay, it has one main root and many lateral roots are coming out, whereas in fibrous root you can see that from the base of the stem many roots are coming out. It doesn't have one main root but many roots are coming out, many of them are having almost equal length, and from there also some many lateral roots will also come out, but it's not that one main root is there. Now this root also, along with performing its main function of absorption and providing anchorage, many other roles are also assigned to root, many other modifications are made. It's not only modification of tap root but many other fibrous roots could also be modified. Now the first important modification is that they are modified for food storage. You would have seen a root of radish and carrot. See, these are basically modified tap root, okay, they are modified to store food reserve, and because of that they are fleshy in nature. Similarly, sweet potato is also modification of adventitious roots. They provide, it's a modification of adventitious root, okay. Turnip is there. Then another role you would have seen in some root is of respiration, okay, they are also aerial roots. See here, they are known as pneumatophores. Like in some plants which are growing in marshy places, like in waterlogged condition, if they are growing, their roots are completely filled with water, means above it is completely loaded with water, soil is there, but still above soil also you have lot of water. So what is the problem occurring here is the gas exchange. Okay, now the roots which are respiring, or the plants parts which are there in the underground portion, they are also respiring and they require gases exchange. They want to release carbon dioxide, they want to take oxygen. So for that process, it becomes very difficult if they are growing in marshy places. Now see, it's a very important thing. Many of you may not be maybe in a confusion that plants are taking carbon dioxide and liberating oxygen. It's not that. Plants are also taking oxygen and they are also releasing carbon dioxide. See, they are taking carbon dioxide during the process of photosynthesis, but plants also need energy. It's not that only just making the food is sufficient. They also need to utilize that food. See, leaves are the only part which are making food, and rest of the part relies on those food and the energy which is present in those food for all their metabolic process. So they will also take their food, they will also perform respiration as any animal perform, means breakdown of food material to release energy is basically the respiration process. So they will also take oxygen, they will break down the food and oxygen will be utilized in the respiration process, and because of that even the roots also they will need oxygen to break down their food and to utilize the energy. So in case of marshy areas, these roots are not able to take oxygen or not able to exchange gases, so they develop aerial roots. You can see here a lot of structures are coming out from the soil. So basically they are coming out from the roots. Okay, the plant is there and nearby like roots are there underground. So these roots are coming above ground. Okay, so they are known as pneumatophores. They are aerial roots. Okay, they have lenticels, lot of pores are there. Lenticels are there which help in gaseous exchange. Now coming to another modification of root is nitrogen fixation. See, plant also require nitrogen. Okay, it's not that they are only performing photosynthesis and synthesizing carbohydrate, they are also synthesizing many other nitrogenous compounds like proteins. Okay, like their nucleic acid is there. Okay, so in nucleic acids you have DNA, RNA. So all these things are also required for the survival. So they need nitrogen. Now from where they are going to get nitrogen? Definitely from atmosphere. You can easily say that either from atmosphere or from soil. Now in atmosphere there is a nitrogen which is N2. Now it's very difficult for the plants to take this N2 and to use them into their metabolic process to make all these components, because this N2 is very having triple bond and it's very difficult to break down. So plants cannot take them directly. So what they do, they take other forms, reduced form like nitrates and ammonia. They take nitrogen in these forms. Now they cannot convert this nitrogen into these forms. See, either if it is present in the soil they can take it, but if it is not present they don't have any mechanism. Now who is responsible? There are many bacterial species which can convert this elemental nitrogen into these nitrates and ammonia. So many leguminous plants also, they contain in their root some root nodules, like some, you can say, some pocket like structures which have a bacteria known as rhizobium, which can perform nitrogen fixation. Now nitrogen fixation is a process by which this elemental nitrogen is converted into nitrate and ammonia, and when this is done the plant can easily take it. So this also role, you can say that nitrogen fixation role is also performed by plants. Then many roots also help them in support, like in banyan tree you would have seen the roots are hanging from they are aerial roots, they are hanging from the tree, and this is to provide extra support for that large tree. Even in sugarcane also you would have seen, say it's just a straight stem. So it has root, but for extra support some roots come out from the above ground portion to provide better anchorage. Then some climbing root like money plant is there, it is an climber, so when it is climbing and when it is attaching it to some support, some roots will come out and they will help them in attaching to that support. Epiphytic roots, like you can see here, they are growing on plants, so they also have roots. These roots we will discuss also, that will have some modification which will help them in survival or some process related to their survival. Now parasitic root example is cuscuta, like this is vanda, okay, and this is cuscuta. In this, parasitic roots are there which help them in obtaining food from the host plant. Then assimilatory root is found in Tinospora. Tinospora is also a climber, and in this aerial roots are found, and this aerial roots perform photosynthesis because it contains a photosynthetic layer, because since it is aerial, in that way it performs photosynthesis, otherwise it is not a very common feature of root. Similarly, in Pinus, in some gymnosperm, it is not very common. In some gymnosperm, some mycorrhizal or fungal attachment is also found in root, which help them in. Similarly, in cycas also some algal means associations are there which help them to perform better. Reproductive roots are also there, like roots of sweet potato. They can also act as bud, and if they are grown, they can give rise to an entire plant. Now talking about root, now they are coming out from apical meristem. See, apical meristem give rise to roots. We have already discussed lot of things about it. See, apical meristem is found here. Now it will give rise to layers, it will give rise to protoderm, okay. Now protoderm will form the rhizodermis. So basically this is the rhizodermis, the outermost layer, or it is also known as epiblema or piliferous layer, because it is quite distinct from the epidermis of the stem, because it is not having cuticle, neither it is having stomata. You will have ground meristem, which will give rise to the cortex, and then you have procambium, which will give rise to the vascular tissues of the root. Now, when you observe this root, the growing tip, however, on the lower side, it will always have a root cap. So, the apical meristem, that also we have discussed in the previous classes, that they are subterminal in position. It is as compared to the stem apical meristem, it will have a root cap, okay, and the center portion of the root cap will be known as columella. So, they will also be formed generally from these apical meristem portion. Now, when we observe it, we can divide this entire root zone into different groups. You will have zone of division, okay, which will contain the growing region, which will contain the apical meristem. It will have apical meristem, okay, and then this will be a root cap zone. Then in this, the tissues will be undifferentiated, they will be smaller in size, they will be isodimetric, they will not be differentiated into different layers of xylem or phloem. So, their structures will be almost similar, and they will be highly dividing cells, okay, they will be metabolically highly active. Then followed by this zone of division, you will have zone of elongation, where you can see the cells will elongate, okay, the cell will increase in length. Suppose you have smaller cells here, then here the cells will elongate, and this is very important zone. Like before entering into differentiation, it is important that the cell should increase in length, okay, and this increase in length is actually responsible to the increase in length of the root. So, this zone is very important, and after that you will have zone of maturation, in which you will see lot of differentiated cells, root hairs will arise. So, once the cells have completely matured and they have attained their full structure and full function, from this zone they will start functioning, and they will give rise to root hairs, and then after that lateral roots will come out. Now this picture will clearly show you that how different layers of these zones are there. You can see root cap here, then meristematic zone is there, that small portion. We have also discussed about quiescent center in the center of the apical meristem. Elongation zone, where you can see the small cells are elongated, but still there is no differentiation. Primary like sieve tube elements have started to differentiate, but they haven't differentiated much. Similarly, vessels have also started. Then when it comes to the zone of maturation, you can see a fully mature vessels with thick walls are there and root hairs are coming, and after that you will see formation of lateral root also. So this also we have discussed that in roots pericycle is the place where lateral root formation takes place. So you can see here that in after zone of maturation, lateral root hairs will be formed. Now the rhizodermis cells which are giving rise to these root hairs are known as trichoblast, and root hairs are basically single cell, okay, and they are just enlargement of the cell, of the outer layer of the cell, which is giving rise to those root hairs. Now coming to the structure of a dicot root, see it is more homogeneous. If we talk about, see it will have, definitely it will have epidermis, or you can say epiblema or piliferous layer or rhizodermis. Now this will have, this is an extension, so root hairs will be there, okay, root hairs will be there. Then it will be more homogeneous, more as compared to dicot, where you will find different hypodermis and sclerenchyma, collenchyma. In roots you will generally find in the cortex all the parenchymatous layer, suppose this is all parenchyma. Then you will see barrel shaped endodermis, means endodermis will be, this is all parenchyma, okay. Okay, so barrel shaped cells will be there. Casparian strips you will find, because generally it is Casparian strip is found in the root endodermis, okay. So thick walled Casparian strips are made up of suberin, okay, strip, they are made up of suberin which are deposited on the radial and transverse wall, okay. So in endodermis you will find thick wall Casparian strips, but along with that there will be some cells which will be devoid of those Casparian strips, and those cells are known as passage cells. These cells will allow easy movement of water, whereas those things which have Casparian strip will hinder in the free movement, and this is only just to regulate the flow of water. Suberin is highly impermeable to water. Then after that you have almost single layer of pericycle. Now pericycle is very important because it has meristematic activity and it will give rise to lateral roots. So you will have pericycle here. Now in this pericycle, in this is actually the steel structure, means the central cylinder, and inside this you will see vascular tissues. Now in this there will be some arrangements of this vascular bundles. Now this will be slightly different from the stem, so we will try to discuss all those things. So, when you will observe the internal structure of this steel, you will see that the vascular arrangement is radial. See, this is very important, which feature of a root, where the vascular, you will have different patches of xylem and phloem. See, the xylem and phloem will not be found on the same radius, but they will be found on different radii. So, this type of arrangement is known as radial. So, here also you will see xylem, this is the xylem, okay, and this is the phloem, and in between xylem and phloem you will see the tissue which is filled is known as conjunctive tissue. Okay. In this also you have exarch arrangement of xylem. So, this is very important, because in this case metaxylem will be in the center and protoxylem will be in the outer side. Pith will be there, but pith in case of dicot will be highly reduced. You can see the area is very reduced. So, in dicot pith will be very small, or it may even be almost absent, and you will see 2 to 8 xylem poles, means these poles of this xylem will be about 2 to 8 in arrangement. Now, this we have discussed, it is in exarch condition. This is also a figure which shows you, see, it's also very important to remember that this passage cells are coming it is opposite to the protoxylem element, okay, where the protoxylem elements are forming, at just opposite to it passage cells are appearing. Okay, so rest of the things have Casparian strip. Now see, it's a tetrarch arrangement where you can see the pith is highly reduced. Okay, you can have different arrangement of this vascular tissue. You can have a diarch condition. Okay, this is a diarch condition. Then you can have tetrarch condition. Okay. In this also you can have phloem here, and up to 8 xylem poles you can see. See, you can see multiple, but it's not so high as compared to monocot, because in monocot you can see still more arrangements. Generally in dicot you can have tetrarch or pentarch or triarch condition is very common in case of xylem. Now this is just a figure of a ranunculus stem to show you that how the arrangements are there. You can see epidermis, then cortical rays are there which stores lot of starch, so starch grains are there. Then you are having an endodermis, and then pericycle is there. In between you have tetrarch condition of metaxylem, and then protoxylem. In between you have conjunctive tissue. So, thin walled passage cells are also there, okay, and they are just opposite to the protoxylem layer. Now, this is also a longitudinal section. Longitudinal section of this xylem contains protoxylem, as we have talked, it is on the outer side. You can see the spiral and annular thickenings in the protoxylem, and metaxylem is in the center. You can have pitted thickening, okay. Along with that you will have various other elements like fibers and trachees and all those things will be there. Then phloem will be there in between. So, okay, so sieve tubes will be there, companion cells will be attached in the sieve tubes. Endodermis, you can see thick walled endodermis on the radial walls because of the presence of Casparian strip. Now, transport, see, root has a very important function of transport of water, and transport of water starts with the root hairs. See, root hairs will increase the surface area and it will help in better absorption of water. See, in the earlier classes we have discussed, while we were discussing about xylem, that how the water will enter into the vessel, okay, this is the vessel, and then it will move upward and following the stem, okay, and because of the cohesive and adhesive properties, adhesive and adhesive properties of water, this overall flow will take place, and transpiration will help in pulling the water from the root to the stem. There will be a negative potential created, and because of that water will move upward. Now in this we will try to discuss that how from root hairs this water will actually enter the vessel. See, this is the root hair, okay, root hair. The root hair will absorb water through the process of osmosis. Okay, then this will enter, it will pass through various cells of the cortical layers, and then it will enter the endodermis where you have Casparian strip also. Okay, which will not allow a complete movement of water, and then it will enter the conjunctive tissue and finally into the vessel. So, how this actually whole water is moving inside these cells, let us try to understand. See, basically there are two pathways of this water movement, or actually in the entire plant also there are two pathways. One is known as symplast, which is followed by plasmodesmata, and the other is apoplast, which is through cell wall. Suppose if we try to understand this process, okay. So, in this diagram, suppose this is the vessel and water is coming out from root hairs. So, they will follow two pathways. One is through plasmodesmata. This is the plasmodesmata, which are the internal connections which are posed, through which substances can move freely from one place to another, from one cell to another, and through cell wall also they are moving. So, when they are moving through this symplastic route, it is okay, fine, they will move freely, but when they are following the cell wall, see, this is the cell wall, when they will reach the cell wall, they will reach the endodermal layer, they will see here a Casparian strip is there. Okay, in the endodermal cells, Casparian strip is there, which will not allow the free movement of water. So, they will have to follow the symplast way only. So, in this way the Casparian strip is regulating and it is allowing only the symplastic flow of movement. Otherwise, through passage cell both the movement could be possible. So, in this way water is entering from root hairs to the vessel. So, with this I end up in this lecture. In the next class we will start with this root section. Okay, we will complete this root, and for today thank you.